Hello everyone, my name is Raphael and I will present the work developed during my Master's at PUC-Rio under the advice of Professor Márcio Carvalho. This research is a 3D visualization of oil displacement in porous media by the injection of microcapsule suspension using confocal microscopy. The main focus of this work was to perform experiments to improve our understanding of some aspects of the oil recovery process. In this way, we focus on secondary and tertiary methods. Briefly speaking, secondary oil recovery consists of the injection of water or gas to pressure the reservoir. Because of the viscosity difference and capillary effects, a large part of the oil still remains inside the reservoir after the secondary recovery. Thus, tertiary methods are used to mobilize the trapped oil. These methods comprehend different strategies, such as the injection of chemicals, particles, and the addition of heat. In this work, we analyze the effects of the injection of an aqueous suspension of microcapsules in order to create blockages in the water paths. This causes the pressure gradient to increase, and if it's greater than the capillary pressure, it mobilizes the trapped oil. The microcapsules injection is similar to the tertiary recovery method using emulsions. When injected, emulsion droplets block the water preferential path, as shown in this picture by black arrows. Then, the additional pressure forces the new path for the water to displace the oil, as shown by red arrows. A field test of this technique was performed by Microlife in the Midway Sunset Field in California in the 70s. The result showed an increase of oil production 3 months after the injection of a batch of 3,000 barrels of emulsified oil in water. After almost a year, other 30,000 barrels were injected. In the end, the oil production was 55,000 barrels higher than the expected without the emulsion injection. When working with emulsion injection recovery, one of the difficulties is to control the mobility properties of the emulsion. Thus, injecting microcapsules could be an alternative to the emulsion injection. The microcapsules used in this work consist of a double emulsion produced using glass capillary devices. This type of microcapsules have controlled diameter and shell thickness. This work is the continuation of Vimiero's master thesis, where he injected PDMS microcapsules in an artificial porous medium. Due to the stiffness of the microcapsules used in his work, they could not penetrate deeper into the porous medium. However, even with that setback, he was able to observe a small increase in oil recovery. In our research, we have improved the visualization technique and tested a softer type of microcapsule. So, in summary, the objectives of this work are two: to improve the experimental technique by achieving 3-phase simultaneous visualization and by increasing the depth of visualization, which allows the investigation of regions away from the porous device walls, and to investigate UR by testing the use of microcapsules injected with water to mobilize oil trapped inside the porous medium. Now, we should consider the review of the experimental procedure. The experimental setup is composed by 2 syringe pumps, the porous medium, and the confocal microscope. The first step involves the oil saturation of the porous medium using syringe pump number 1. Then, we change the syringe in pump 1 and start injecting the aqueous phase. After the injection of 10 porous volumes, no more oil is produced. At this point, we inject the aqueous phase with the suspension of microcapsules from pump 2. It is then observed an increase in oil production after the microcapsules injection, with an extra amount of oil leaving the device. A 3D image is generated after each step using the confocal microscope. Those images took approximately 3 hours to acquire and were used to analyze the oil displacement. In this presentation, we will show only the result obtained with the one-sized glass sphere devices, with 425 microns. The porous media have a theoretical pore trough size between 180 to 207 micrometers, while the capsules have a diameter of 104 and a shell thickness of 5 micrometers. We have also performed some experiments with different porous devices, composed by layers of spheres with different diameters. The microcapsules adopted were prepared using a co-flow and flow-focusing capillary device. As mentioned before, microcapsules are double emulsion, in this case, oil in water in oil. The composition of each phase can be seen in the left-hand side of this slide. At the right-hand side, a diagram presents the concept of the capillary device. Below that, there is a video showing the microcapsules production, with controlled diameter and shell thickness. Regarding the visualization technique, we use confocal microscopy. We know that confocal microscopes are commonly used in the biological area in order to analyze 3D samples. That is not achieved with common microscopes that need a cover slip. This comic strip explains the advantage of the technique, allowing 3D visualization and even a dynamic analysis of the sample. In order to produce the 3D visualization, this equipment relies on pinhole filters, which restrict all the light from out-of-focus planes and let only the focal plane light to pass through. So, each section of the sample is acquired separately, and later reconstructed to form a 3D image. Fluorescent pigments are added to each phase of the sample and are excited by a specific laser beam, emitting in different wavelengths. The emitted light is then captured by using PMT or hybrid photodetectors. The procedure for the 3D reconstruction can be better explained with these images. First, a stack of 2D images of different focal planes acquired from multiple positions. Then, each stack is rebuilt in 3D separated tiles. Finally, these tiles are merged together to represent the whole sample. The aqueous phase is shown in blue, the oil phase in yellow, and the capsules in purple. It's important to note that the black areas are the glass beads, which don't emit any fluorescence when excited by the laser. As mentioned before, the artificial porous media used in this work are composed by a package of glass beads, sintered in a furnace at 875 degrees Celsius. It is very important for us to match the refractive index of these glass beads with those of the fluids in order to minimize inner refraction and to improve the depth of visualization. As mentioned before, the artificial porous media used in this work are composed by a package of glass beads, sintered in a furnace at 875 degrees Celsius. It is very important for us to match the refractive index of these beads with those of the fluids in order to minimize inner refraction and to improve the depth of visualization. The porous media are placed in a square pipe of 2 mm side. Choosing the fluids for the experiments demanded the miscibility of the oil and aqueous phase, the matching of their refractive indices, and a proper viscosity ratio between the phases. Because the phases have the same refractive indices, the confocal technique needs the addition of a fluorescent pigment to distinguish them. We have carried out a survey on fluorescent pigments, which allowed the simultaneous visualization of oil phase, microcapsules, and aqueous phase. The final part of the work aims to develop an image processing procedure to quantify the experiments. To do so, we have used two computer programs, ImageJ, an open-source software to pre-process, segment, and post-process the image, and the ORS Dragonfly, which has more resources for the 3D analysis than the previous one. On the right-hand side, it is possible to see a single slice before and after the segmentation. Segmenting the image means distinguish different phases in it. Let's take a look now in the experimental results. As I mentioned previously, the first step of the experimental procedure is to oil saturate the porous device. That becomes the initial state of the system. The results are presented single-plane images to allow easier visualization. Along with the porous device properties, the color used for each phase is shown in the top corner of the following slide as a reminder. As expected, we can observe that the higher the oil injection flow rates are, the higher the oil saturation is. Some regions, highlighted in red, show pockets filled with the aqueous phase that are displaced by higher oil flow rates. A 3D visualization is also possible, allowing the analysis of the residual aqueous phase. The images of this step also show that the wetting pattern of the glass beads. After the device is saturated with the oil phase, we start the aqueous phase injection. As the aqueous phase flow rate increases, the following aspects can be observed: less oil trapped in the porous medium, and a large number of smaller and separated oil ganglia. These observations are in good agreement with the work of Janunananda and Morrow and the work of Zhao et al. Then, when no more oil is produced from the porous device, the microcapsules injection starts at a constant flow rate. It is possible to observe interesting effects of the microcapsule in these 2D images of a single slice, before and after the injection. The aqueous phase preferential path is blocked by some capsules, as shown in the purple circles. Also, red circles in both figures show the movement of the oil ganglia inside the porous device, as a result of the microcapsules action. Another interesting result is observed at the exit tube. Shortly after the microcapsules injection, it is possible to see oil production in the naked eye. Summarizing, after running many experiments, it is possible to conclude that the microcapsules work in this artificial porous device as a method to displace additional oil that was trapped before, break oil ganglia into smaller ones, and even in ganglia that are not fully mobilized, the increase of local pressure causes small deformations. In this work, I could improve the confocal technique to achieve a good resolution. The first image on the left shows we can visualize the details in 3D images. Also, for the picture on the right hand side, using 3D printed design holders, we can rotate the porous device on the microscope. That allows the visualization of both sides of the device. In this way, it was possible to reconstruct and analyze the whole sample. And, as will be shown in the next slide, it is possible to perform a dynamic visualization of the porous region. In this sequence of acquisition, it is possible to observe the presence of a microcapsule, marked in yellow, suffering pressure deformation as the flow rate increases. Then, another microcapsule passes and blocks an adjacent pore. After a short time, the second capsule is forced downstream. The time between each frame is 15 seconds. Finally, the last part of this study is to show that it is possible to quantify the phase distribution in the porous media using 3D images, plane by plane. Some techniques are used to reduce noise, such as filters and phase subtraction. The picture on the left shows the resulting image after this process. The table displays the result of one experiment where the increase in oil recovery can be clearly seen. Just minutes after the beginning of the injection of an aqueous microcapsule suspension, a sensible drop of nearly 7% in the oil phase saturation is measured. A small additional oil recovery is observed by continuing the aqueous phase injection up to 10 porous volumes. Then, what can we conclude? In this project, we have improved the technique of 3D visualization of porous media flow using confocal microscope. We have achieved imaging the 2 mm square device in its whole depth, capturing 3 distinguished phases. We have observed that the microcapsules could penetrate the porous medium and block the aqueous phase preferential paths. This blockage has had an effect on the trapped oil displacement and mobilized part of it, so we provide a proof of concept that supports EOR using microcapsules. Finally, it is possible to visualize the increase of oil recovery effect by the observation of oil passing through the exit tube. This fact is also supported by the saturation data provided by image analysis. The relevance of this work is to provide data and information for further research on microcapsules injection as a way to improve oil recovery. At last, there is still some improvements under development in this work, such as the measurement of device pressure drop before and after the microcapsule suspension injection, and the analysis of volume and distribution of each ganglion. This work has counted with the support of Shell and of the Brazilian National Research Council, CNPq. Thank you for your attention.